The need to analyze proteins in single cells has been appreciated for decades. And Dr. Bendall has been instrumental in establishing one of the earliest high-throughput methods for simultaneously characterizing over 40 cellular parameters across many single cells, namely mass cytometry. He was first author in 2011 of one of the most influential papers on the subject, and he's been extending the applications of the platform ever since. Using this technology, Dr. Bendall has identified rare pro-inflammatory subpopulations of chondrocytes in osteoarthritic patients. His investigations of acute lymphoblastic leukemia uncovered cellular signaling features predictive of disease relapse. And his characterization of immune cell signaling pathways in post-surgical patients demonstrated the efficacy of CyTOF-based methods for predicting patient recovery. This afternoon, he'll be discussing immune cell metabolism and its relationship to disease states. Let's welcome Dr. Bendall.
Maybe that's better. My background, really comes from a de novo mass spectrometry-based proteomics world, where I got my start in a lab working on a multi-TOF DESTR doing peptide mass fingerprinting. And it was really my work in stem cell proteomics that kind of inspired me to pursue single cell proteomic approaches, which at the time back in, it was like 2007, 2008, it almost seemed like a bit of an oxymoron. And, I think as all of the work that's been talked about today already, and it's coming up tomorrow, this is rapidly changing. So to just kind of, I'm going to give you a little overview of the technologies which were already introduced, and then tell you a little story we're doing to kind of get at single cell metabolic measurements or approximations, and how we're using this to study the immune system and disease. And when we think about these single cell systems that we study, I think there's different ways to think about how we use single cell data, and that somewhat informs some of the approaches that we've taken here, right? So why do we like single cell data? We like single cell data because it gives us multiple levels of biological organization, right? We've got this kind of in silico organization in the top right here, where you have this system that we're kind of in silico, we can create a hierarchy and understand how cells are related to each other. Of course, actually in vivo, in multicellular organisms, cells actually organize into structures, with these structures down below here in tissues have patterns, and those patterns are meaningful, right? The localization of these cells is not random. And of course, all of this is driven not just by proteins and glycoproteins and molecules on the surface that we use to kind of phenotype cells, but also kind of the molecular information, the organelles, the transcription factors that are intrinsic to inside the cell and how they're organized and their expression levels dictate cell function. So the way the single cell proteome approaches I often employ work really relies on these kind of we'll call them old school targeted approaches that rely heavily on affinity reagents. So this idea that you can use antibodies or small molecules, nucleic acid probes, things to kind of bind the target you're looking for, right? Because really up to this point, particularly in a lot of realms like immunology and hematopoiesis, you never measured the thing directly in the cell that you were looking for, right? We just didn't really have the tech there. And so what we would do is we'd probe for it with an affinity reagent. We'd attach something to that affinity reagent, like fluorophore or an enzyme, that we'd read out that we could detect the activity for, and we'd read that out as a surrogate for the actual thing we wanted to measure. And so right as when I was starting my my postdoctoral work to kind of look at these kind of single cell proteomic approaches or single cell biochemistry, an advent of technology came up where a group in Toronto led by Scott Tanner got this idea that you could actually swap out these fluorophores or enzymes and use non-biological elemental mass reporters in the place of these typical fluorophores or enzymes. And this gets around a lot of the signal overlap you can see here. It allows a huge amount of multiplexing in addition to what you could normally get with fluorophores or enzymes, as well as, you're all familiar with mass spec technology, mass signals, difference between a a proton or a neutron, you can drive a car through it, right? So this is relatively easy. You don't get signal overlap. You don't have to compensate, as well as you get all of the kind of sensitivity and dynamic range that's inherent with mass spectrometry ion counting. So the basic workflow for single cell proteomics by CyTOF mass cytometry and very similar to the tissue imaging approach I'll also describe is this idea is you have your cells, typically they're in suspension. The suspension, often these cells can be from different states, either drug treatment states, disease states, time points. That's kind of the biology end of the assay. We then freeze these, often chemically in time with fixation or alcohol or temperature. And then we come in with our various probes. Again, like I said, mostly monoclonal antibodies, but pretty much anything we can conjugate one of these metal chelators to. Once everything's stained using pretty standard hybridization and immunochemistry type techniques, in the case of the CyTOF, a suspension of cells is then sprayed into the mass cytometer, where the cells are physically separated in time, where there's really only on the order of microseconds separating each of these droplets. Each of those droplets is vaporized, atomized, and subsequently ionized to their atomic constituents. Those constituents are measured on the order of about 80,000 times per second. And then you can see a Gaussian come up of each single cell, and all of that's rapidly integrated and quantified, where you end up with a single file per cell with the integrated amount of each of the isotopic constituents, which relates to the overall level of the different molecules you're actually probing for. And importantly, and I think this is important when we're thinking about like growing this community of single cell proteomics, it's important to couch these things, right? So everything that I've heard today has talked about a lot of depth. You guys are going way deeper than we can go with this, right? But what I can say we can do is we can really sensitively probe routinely 50 things or so in a cell in a targeted fashion. And some down to the level of 10, or even in some cases, single molecule sensitivity. And with this type of assay, we can measure millions of cells in a single day on one instrument, and we can get relative linear quantification across about 4 logs of all of those targets. And all of this can be done if you amortize it to, the number of cells you started with versus the number of events you get out for a fraction of a penny per cell. So, this is the type of technology where even though we can't go super deep, what we can do is we can look at a lot of cells, which means we can look at systems that are complicated with rare cell types and build complicated models. And here was an example of some of my early postdoctoral work where we took the human hematopoietic system from bone marrow, and we could capture, rare, less than 0.1% of the system hematopoietic stem cells, all the way with super abundant, like T cells and B cells, which can be up to 70% of the system. We can identify those with part of the panel, and then we can start to ask functional questions about those cells by probing with immune signalers like IL-7 or other receptor cross-linking, and then we can look at one condition versus another and figure out what cells are responding to these different perturbations, whether it be a cytokine activation, lymphocyte receptor cross-linking, or something like a toll-like receptor agonist like LPS. But this can also be done, like as I alluded to, in the context of tissue. So another embodiment of this method is we were also able to go and apply this to tissue sections by taking the similar assay with the non-biological elemental reporters and combining it with a type of mass spectrometry called SIMS. In particular, this initial embodiment was done on an instrument called a Kamiga nanosim. So this SIMS is essentially a spatial scanning mass spectrometry. It's a little different from MALDI in that instead of using a laser to do spatial sampling of your tissues, it uses a charged primary ion beam. And this primary ion beam can be focused down to a nanometer scale spot size, and it can raster quite rapidly across the sample. And as it's rastering, for these tissue sections where we've stained with antibodies, the reporters are freed, and those reporters can be spatially quantitated to the point where we can build up these elemental maps with nanometer scale precision and map out protein expression in like breast carcinoma, as we did in this first example. The issue, of course, if you've ever seen a Kamiga nanosims, is a very scary instrument. The instrument it's about $5 million. It takes about 6 to 12 months for it to even be installed in your lab. It actually gets built inside your lab because it's so large, about the size of a conference room. It is extremely complicated. I've heard people who use it talk about, they're like if they were a pilot, they figured it would be like flying a Boeing 747. And so it was great that we were able to kind of get this proof of concept done, but one of the things we realized very rapidly is that if we wanted to use this technology more routinely, much more like we use CyTOF mass cytometry for immune profiling, we had to come up with a different format that was purpose-built. And so in this case, we've recently, and I just wanted to kind of cover this briefly because we use the technology in the main story I'm going to be talking about today. We built an entirely new set of instrumentation, working with some consulting firms and some engineers in our lab at Stanford. We built a multiplex ion beam, or MIBI TOF, time of flight detector prototype in the lab that we've been using for larger scale pathology studies. And so what we did here is we kept all the things we wanted about SIMS, right? We kept the primary ion beam. We actually juiced that up a little bit and made it bigger and brighter so we could blow more stuff off the tissue more quickly. And we took out the magnetic sector, which was the original detector, and we put in a time of flight device, and that let us do the parallel mass measurement. And then we added some optics in between to kind of filter out the ions we didn't want, like the multi-atomic species, and keep the ions we do want, like the monatomic species. And you can kind of see that here. So again, this nanometer scale beam rasters over the tissue sample that's been stained with all the different metal conjugated probes. You get a variety of ions being sputtered off. Some are low energy clusters, some are high energy monatomic species. The low energy clusters kind of, it's like your tire getting stuck in the sand. They overturn the corner and they get filtered out here. And the monatomic species go through to be rapidly quantified and identified. And again, this same instrument, the TOF is scanning on the order of about 100,000 scans per second. And the beam is rastering at about, on most of our experiments, around 1,000 pixels per second. So you're getting about 10 TOF measurements per pixel, which gives us lots of signal on a pixel by pixel basis. And the long story short is what we were able to do here is we were able to go from this initial experiment here on the NanoSIMS in 2014, where it took us a couple hours to measure 7 elements across a window of this large, to where we can now scan a tissue section this large at equivalent resolution in well under an hour. In this case, this actually was about 17 or 18 minutes. So we can do really large scale full slide scans, or we can do studies where we're measuring fields from hundreds of patient samples per study. Some of the cool stuff that also comes up with something like multiplex ion beam imaging is this idea that we can look at these non-biological elemental reporters. We can also look at endogenous elements as well. And there's lots of, as everyone, this crowd, I don't need to tell, there's lots of interesting things about endogenous molecules and biological material. Everything from looking at carbon and phosphorus, which, in this case is kind of boring because we're just looking at the native stuff, but you can see carbon kind of matches the skeleton of the cell outline. Phosphorus, you can pick up the DNA and the nuclei of the cells. You can get a little more creative, and we can go to all these old school pathological chromogenic dyes, most of which were heavy metal, transition metal based. So something like uranyl acetate does a really nice job of staining the walls of these of gut epithelium here. Or you can go and look at metals that are actually involved in key biological processes. Something like iron is picked up by tissue resident macrophages when they're scavenging red blood cells during kind of tissue maintenance and homeostasis. The other cool thing is SIMS is quite precise in the amount of energy that's delivered to the tissue. So you basically can use the particle beam as a pseudo-confocal approach to kind of raster down through the tissue section and create a Z stack of the image. And so these are even some examples where we don't do it a lot because most of the time the data is already complex enough in 2 dimensions. We can go through and we can actually kind of create a 3D rendering of a tissue section. So that's just a little background on kind of the single cell droplet profiling techniques and the tissue imaging and analysis techniques. And today I'm going to talk to you guys mainly about one key thing, which is something near and dear to my heart and in the minds of most folks in my lab, which is kind of new assays, new questions. What are new things we can use this toolbox to learn about biology? And so one thing we had been really trying to get at for a while was this idea of cell metabolism, because metabolism and how the cells are, leveraging energetic processes to carry out different functions seem to be so intrinsic to cell identity and cell function. And obviously is something we know in a number of scenarios gets perturbed in disease. And all of this work has been led by Felix Hartmann, very talented postdoc who took a risk on coming to a new professor's lab. And I think it's gone really well. He's going to be on the job market pretty soon. So there's his info, and you can contact me later if you want to be put in touch with him. But he's a great guy, and I will be sad to lose him at some point soon. So the idea of metabolism, again, it is you can go and you can read Cell, and there's these nice review articles where we see the tumor microenvironment, and we can see, oh, there's tumor cells and immune cells, and there's metabolites flowing back and forth. But it's really hard to actually get a true picture of this, right? These are all estimates of what we think we can go on, because we can only take small slices of these systems for analysis. And I think the important part is it's not, even though I think a lot of the de novo approaches for studying metabolism typically look at the metabolites themselves, often leveraging techniques like mass spectrometry. I think the other side of the equation is all of these processes are driven by regulatory enzymes and transporters and a lot of kind of physical structural pieces as well. I think the other thing that, of course, that creates a really challenging scenario when I talk to people who study metabolism is this idea of kind of sensitivity of detection, and how do we, much like with proteomics and what everyone's talking about here, how do we integrate these measurements into our cellular systems where we can get or approximate single cell information, because we know cell A and cell B aren't necessarily the same cells, so we don't necessarily want their metabolites or metabolic pathways being mixed together. And so you've got this sensitivity issue, and I think probably the part I really didn't appreciate until we started looking into this more is there's also an analysis kind of well, it's the Schrodinger's cat problem, right? If you open the box, you perturb the system, and so you don't know if what you're seeing is kind of true anymore. So this idea that, particularly for the immune system, if you go and actually take your cells out of someone's body or out of the animal and put them on a cell sorter and purify them into a well to then go and do some downstream assay, you've actually perturbed the cells, you've changed their osmotic pressure, and you've changed their metabolite concentrations. So how do we solve this problem? Well, turns out, in our case, we have a really good system, quite a sensitive system for doing targeted measurements of something. So what can we target, and what are the components of the system we think are relatively stable? So the analogy we wanted to take here is kind of this idea that people look at the central dogma from, right? We've got our kind of chromatin regulatory regions, right? Genes are either open or closed. We often then, if they're open, then they can be transcribed, and you've got your RNA. And of course, if the RNA is doing all the right things, then it can be translated. It makes the workers of the community the proteins, right? So if you look at a metabolic pathway, I kind of think about the metabolites as the RNA, and kind of like our field, right? A lot of people have the misconception, or maybe not, I wouldn't say misconception, but certainly it's easy to make the assumption that if you measure the RNA or you measure the metabolites, you're capturing the whole system. And then in a lot of cases, there's a lot of information, and a lot of that is true. But I think what we did is we had this idea that if we kind of measured the orthogonal component, in this case, the go-betweens, the enzymes that were regulating the transitions between these different steps, that the enzymes might serve as a reflection of the metabolic state that was going on. So this was our big assumption. And working from that assumption, we then went in and we started to kind of in a somewhat manual, educated, biochem 300 driven fashion, we went down and we tried to start kind of tearing things apart here, where we went into kind of the major metabolic pathways, and we asked, well, what are the rate limiting kind of enzymes and transporters, responsive transcription factors, regulatory modifications that we think are kind of indicative of these different pathways and thus these functions. And so we developed metal conjugated probes against all of these things on this list, as well as screened a whole host of other material. And then we were able to take them and put them all back together into a multiplex panel where we could still incorporate other facets of cell identity. So we could still phenotype the cells, we could add in facets of cell cycle and other functions of cell behavior. So we could still do all the same assays I talked about before, but now we could add a layer of metabolism into all of our questions and answers. And you know, you don't need to, if you're writing a lot of this stuff down, all of this is actually all 112 metabolic targets that we screened and tested for both the cell suspension and the tissue-based assay are all part of the publication that's currently online and bioRxiv, as well as coming out in print pretty soon, hopefully. So how do we kind of initially validate this? So to let you understand the context here, we were coming from, you know, kind of a systems immunology background. And in immunology, probably one of the best characterized processes in immunology in terms of metabolism is cytotoxic T cell activation. And so what we wanted to do is not break something that's already established. We wanted to kind of come at this as our first pass for kind of benchmarking our analysis system. So what we did is we purified primary human T cells in a dish where we could then activate them using standard procedures, receptor cross-linking, the same sorts of beads and molecules that people use to generate and expand and make therapeutic T cells. We were basically doing this to make research T cells. And at every day we would take an aliquot out and we would have that as an analysis time point. And what we did with these time points is we benchmarked them using metabolic flux analysis. This is an instrument that Agilent sells. It's called the Seahorse. Essentially, it has two sets of reagents or two kits that let you quantify both glycolysis as well as aerobic oxidative processes. And so we were able to generate profiles of our cellular activations over time using both of those platforms, as well as apply our multiplexed single cell metabolic probes. And so here's just showing you up here top two glycolysis and fermentation probes, as well as one probe for the TCA cycle and one probe for mitochondria and the electron transport chain. You can see as you go from 0 to day 3, you get this nice T cell activation and shifts in both of those pathways. But more broadly, you can see this happening across multiple molecules along those pathways where everything's generally moving in the same pattern, but there are some differences from molecule to molecule, which we'll go back into, which is sort of interesting. But of course, then we can go back and we can compare this on a one-to-one basis with either the glycolysis score or the respiration score that you see off of the Seahorse. And on these bulk assays versus the medians from our single cell datas, we were able to get very nice one-to-one correlations across all of the time points. But of course, you know, it's nice to be able to compare the medians and kind of show concurrence. But again, all of this was actually single cell information underneath, where even at any given day, the cells were actually covering a spectrum of behaviors. And those behaviors were dynamic, and of course they were dynamic across multiple pathways as well as multiple facets of cell kind of functional behaviors. So we had all of these cell cycle measurements as well as measurements of biosynthesis here, which is DNA replication, transcription, and de novo translation. And so what we were able to do is then kind of getting past this benchmarking state, we were then able to go and kind of use some of our single cell tools. And whenever we're following a dynamic system, one of our favorite tools to kind of break out is pseudotime, right? Where this idea that like, you know, well, there's kind of a median at day 0, day 1, day 2, and so on. There's always a spread of cellular behavior, right? You can see day 1 occupies multiple states, day 2 occupies multiple states. So what we do is we pool all the single cell information and then we reorder it using kind of the phenotypic and functional domain of all of the parameters that we've quantified. And in doing that, we can then kind of build this dynamic snapshot of what the system's doing over time. And we can create a trajectory through that system and then use it to map the ordering of all the processes, which is what we did here for the cytotoxic T cell activation. And so what's kind of cool here is you can break this into these phases, which were kind of already understood, but typically understood from much more complicated experiments than this one that really was a bonus out of a validation experiment. So you see initially you get T cell activation. Once the T cells are activated, they enter something what we call the acceleration phase, where you see a lot of the metabolic enzymes ramp up, including a ton of de novo transcription and translation. So their cells are bulking up, they're building more proteins, and then eventually they get into cell cycle entry. Once the cell cycle is on, you're at your kind of peak metabolic state. And what's interesting here is we see this at the metabolism level, but we also see this at the cell phenotype level in a previous study, which is as soon as the cells divide for the first time, which is indicated by this last maroon line, you get this wholesale switch in cell identity. It's really this first switch, and this is something we kind of understand in stem cell and developmental biology. Cells have to divide to differentiate, right? So they undergo this first division, and that's when they've really kind of locked into their cell fate and made a new choice. And of course, those new choices not only come along with new identities, but new metabolic wiring. A couple things that are kind of interesting about this in terms of a technique for monitoring cell behavior and monitoring things immunologically is that, you know, this is relatively robust across multiple cell types, irrespective of how those cell types are collected and stored. This is looking at the immune system, but this is really important because certainly things like a lot of PTMs like phosphorylation or a lot of things like RNA aren't very stable and are very sensitive to degradation and changes with storage collection conditions and collection. The other thing that's interesting on a biological standpoint, if I take a whole bunch of replicate blood draws from a bunch of different individuals and run the same panel, what you can see is if we embed the median data from these individuals just on something like principal component analysis, cells that are phenotypically similar and thus presumably functionally similar embed in the same place. So they have very similar basal metabolic wiring, where very different cell types like your dendritic cells versus your lymphocytes, which are all similar here, they're quite far apart because they're wired very differently. So this idea that cell identity and thus function is intrinsically tied to cell metabolism, at least in the resting state here, seems to be true. And this we were able to show extends across multiple tissues. And so what we did here is we not only looked at those 5 peripheral blood mononuclear cells, so this is kind of your white cell immune content. We took lymph nodes from healthy individuals, and then we had paired healthy colon and adjacent colon carcinoma, where we extracted the immune cells from there as well. And we took all of those samples, and we ran our single cell metabolic profiling on them. And the result, a subsampling of the results was all embedded here. And what's kind of cool is, again, this just reinforces even across different tissues, across different individuals, like embeds with like when it comes to main cell functions, right? Your epithelial cells here, B cell island, T cell island, so on. But you can get a lot more granular, right? So we can go and we can select just the CD8 T cells from here, and we can kind of re-embed them and stretch them out just based on the metabolic properties and a little bit of the phenotypic properties that we measured. And you end up with, again, in an unperturbed state, just kind of coming from different tissue locations and different disease scenarios, you end up with all of these different metabolic flavors of cytotoxic T cells. And this is particularly interesting, especially when we start thinking about colon carcinoma, because if you're not a T cell person, your CD8 T cells are those T cells that are supposed to be the ones that go in and they kill tumor cells, or they kill cells that are infected with viruses. They're really the ones that go in and they take out the things that aren't supposed to be there directly in the immune system. And so it's interesting that even in basal scenario, we see these things wired so differently. And so what can I tell you that's interesting about the wiring here? Well, first off, again, just kind of hitting this thing home that intrinsically the wiring is relatively consistent in healthy scenarios. If you look at peripheral blood, or you look at the healthy colon, we see remarkably consistent composition of the metabolic clusters in the cytotoxic T cell states between different individuals, which is each column here. And what's interesting is there is certain clusters, particularly this one, cluster 3, which is one of our little vignettes here I want to talk to you about, that come up only in the disease scenario, and it seems to come up variably in the disease scenario. So cluster 3, the colors match here. This maroon cluster is up here in the UMAP. This cluster is generally metabolically suppressed. So it's downregulated a lot of these enzymes, a lot of these key regulators, except for it's upregulated amino acid metabolism. It's got really high levels of this CD98 transporter. And again, as you can see here, across just the limited number of individuals we looked for in the study, this unique metabolic cluster was there in all of those disease individuals, again, but not in their matched adjacent healthy colon tissue. So kind of interesting. So what else can I tell you about these cells? So these cells, besides being metabolically suppressed and just having overrepresentation of 98, which is that amino acid transporter, the cells are also not proliferative, which of course is not a desirable property. You want your CD8 T cells active and killing when they're in a tumor. They're lacking the T cell self-renewal factor, TCF1, but they have increased levels of these two checkpoint molecules, PD1, which a lot of people have probably heard. This is one of the main therapeutic targets of checkpoint therapy, but they also have this other kind of newer checkpoint molecule, CD39, which serves to decrease the availability of ATP in the microenvironment. So what else can we figure out? Well, what we were able to do now is we can we've learned as much as we could learn from the single cell suspensions, and we can now kind of switch gears and go from the cells in suspension to the cells in tissue. And so we're able to take not the exact same patient samples, because we didn't actually have those same patient samples in tissue section, but relatively similar colon carcinoma samples. And we're able to take most of the same reagents we were using for the CyTOF assays and re-employ them into our imaging assay. And so using that same workflow that I talked through at the beginning, we're able to profile a number of these patient samples. And we were able to kind of recapitulate some of the main themes that I talked about in terms of single cell metabolism in the first part here, which is this idea that, of course, different phenotypically and functionally distinct cells have very different phenotypically and functionally distinct metabolic profiles, right? And you can see that here where the cells kind of segregate from one another. The other thing that was really interesting that we can now answer once we're generating this kind of in situ cellular information is we can start to ask questions about spatial relationships, right? Is there, like in that original cell review I showed at the beginning, is there kind of metabolic neighborhoods? Are things kind of dictated? Do two cells that maybe don't have the same identity but are next to one another have similar metabolic behavior? And the answer to that is actually yes, which is reassuring, but it's also kind of surprising, right? It's this idea that seemingly the microenvironment can play a really important role in that. When we do this independent of cell identity, what this map is showing is essentially Z-score enrichment on a cell-by-cell basis, right? So what we're saying is, like, in a given neighborhood of a cell, if a cell expresses molecule X, you know, what pathway is likely to be on in the cells around it. And so what this is showing is that essentially glycolysis is enriched with glycolysis, you know, TCA is enriched with TCA, amino acid is enriched with amino acid, and this is irrespective of cell identity. This is just looking at cell types across the tissue microenvironment itself, which was kind of cool. And another way to kind of visualize this is we can actually kind of overlay these kind of, I call it like a pseudo PET image. So what we can do is on a pixel by pixel basis, independent of the cells, we can create like a glycolytic score, essentially predict how much glucose usage, for instance, the cells would be using. And what you can see here is, of course, infiltrating immune cells, you would expect them to be activated and highly glycolytic. You get a lot of anaerobic metabolism in those cells. But here's the tumor immune border in the white line. You can see the very adjacent tumor cells, even though they're very, very different from the immune cells, are also highly glycolytic, that are abutting up against those activated immune cells as well. So that was kind of interesting. Now, what about those weird cytotoxic T cells? We got to close the loop here that I talked to you guys about at the end of the suspension part of the story. So we went back and we kind of drilled down on our CD8 T cell fractions within that we were able to image and segment and extract from the tissue images. And we clustered them using similar approaches that we did before. And again, what we were able to see from these images is that there was a large swath of these CD98 high, CD8 T cells, and a subset of which were also metabolically low. So we were looking for the ones that had CD98, but like on the CyTOF data, we're lacking a lot enrichment, a lot of these other metabolic enzymes associated with other pathways. And when we started to ask questions about what these cells are doing or where they are in the tumor, because again, this data was really recapitulated what we saw in the suspension data. They had high levels of checkpoint, they weren't proliferating, they just seemed to not be, they were there and they weren't doing anything. But the other thing that was interesting, and this was relatively underpowered, but we still saw a significant association, is these metabolically low CD98 high T cells seem to actually be, in a non-random fashion, disengaged from the tumor. So wherever we found them in blue here in all of our image, they were much further away from the tumor immune interface than, or from the tumor cells themselves than the red kind of metabolically high, we'd say good-looking CD8 T cells in these tumors. And so again, this idea that probably the metabolic environment the cells are in that's likely related to the metabolic, the metabolite availability probably is playing a role in influencing these cells to become perhaps dysfunctional in the tumor microenvironment. So I think, hopefully I've convinced you guys in this short time, is that using the targeted toolbox like this, which I'm hoping will only get better with some of the de novo techniques that everyone's been talking about here, that we're going to be able to do a much more sophisticated job of proving or disproving these models of how proteins and cells and metabolites and other things that can't be measured with a sequencer all come together to kind of form tissue function and dysfunction. So again, hopefully I've convinced you guys that you can probe the metabolic regulome instead of the metabolites themselves to get yourself a caricature of metabolic wiring in these systems. And by doing so, you can access single cell information. And hopefully if you are into doing kind of clinical profiling or retrospective analyses, you appreciate that we were able to do a lot of this with standard human clinical blood specimens, as well as go back and use archival FFPE tissue blocks that weren't stored in a special sort of way. And that, it seems, at least in our work, focusing mainly on the immune system, that the metabolic state seems to be intrinsically associated with cell identity. The metabolic state gets reprogrammed or rewired with dynamic immune processes, and certainly independent of cell identity, well, there is a strong association, there's also a strong association with microenvironment itself, and likely that probably has to do with local concentration of metabolites. Lots of people, lots of funders. Felix is the star of the show for the single cell metabolism, but it's been, it's been really fun doing all this work and really fun to be part of the single cell proteomics community. And if we've got time, I'll happy to take some questions.
So thank you so much for a fascinating talk. I mean, the ion beam technology was very new for me personally, but to see the advances in characterization of a tissue sample, it was just absolutely floored me. So we have a couple of great questions down here. One from John Yates. Are we at the limit of elemental reporters?
Yeah, that's a really good question. So no is the short answer. So all the best part about this technology, as I've presented it here, is we can, in my lab, routinely multiplex. The exact number is 53 targets. And those 53 targets work with one chelator technology. So I have essentially one label or one backbone that we can load 53 different isotopes into. If you're thinking about one isotope to one reporter, there's approximately another 40 or so non-biological elemental isotopes you could still use, but those are going to require additional chemistries to link them to whatever probes you're using. So that's kind of level one. Level two, which is something that it's in my bucket of crazy ideas, and I don't know if I got to go on sabbatical or I got to find a really ambitious postdoc or something, which is to do the same thing that people do in genetics, which is we start barcoding, right? And I think if you start thinking about, fixed combinations of these things, you can start to theoretically get your tag limit way, way, way up. But that's going to require both a lot of physical work as well as a lot of kind of in silico work to make it all make sense.
Thank you. We have two questions from Professor Slavov. How do you evaluate the stability of your single cell metabolic profiling?
So the stability in our case, I mean, there's lots of different ways we could measure the stability, but in our case, what we did is for this particular paper, we tried a bunch of different collection and storage protocols over time. So fresh cells versus fixed cells, frozen cells versus cells at 4 degrees. And then what we essentially did is across a bunch of different cell types that we showed here and all the different kind of probes that we were using, we looked for a stability of signal. So in this case, it was actually pretty hard for us to find conditions under which our signal would destabilize. And I think that's just a tribute to the fact that we were looking at the proteins themselves and not their modifications, not the metabolites, which can continue to break down and diffuse out of the cell. When you're looking at the metabolites themselves, it's very precarious, right? Anything you do, any buffer you change, all of this stuff goes all over the place. And I think this was why we were excited to kind of get away from going after that, though at some point we need to go back to it. So one of the studies we're doing right now is we're already starting to take this approach and then take a serial section and go back at it with like imaging MALDI techniques and other de novo MS techniques where we can actually measure the molecules themselves properly. Maybe not with the same resolution or sensitivity, but still we can start to look at these kind of metabolic neighborhoods.
And then another question from Professor Slavov is, what affinity reagents can you use with metabolites?
So the metabolites themselves, I would say there are cases where the metabolite's large enough, you can use like antibodies themselves or hapten probes. They're big enough to kind of, you think most binding sites for antibodies tend to be on the order of, 4, 5, 6 amino acids. So they have to be kind of, up close to a kilodalton, kind of half to 1 kilodalton at least. For the smaller stuff, probably the best way to go at it is using a labeling technique, right? The same way we use isotopic labeling. So in this particular paper, we used IDU, so iodinated deoxyuridine, to look at DNA synthesis, and we just measured the iodine, and BRDU, where we actually used a monoclonal antibody against the brominated uracil to get at the RNA. Sorry, not BRDU, BRU. And in the other case, the other metabolite we measured was puromycin. So we used puromycin to kind of pulse de novo translation, and we measured the puromycin with monoclonal antibody. We're starting to play around with doing like carbon-13, deuterium labeling, and N15 labeling, where you can measure individual molecules that you start with, the same way people are doing dynamic metabolite profiling experiments, and we can pick a lot of those things up by the multiplex ion beam imaging.
And then when I was looking through your work to kind of approach the introduction, I noticed that you walk this amazing line between both fundamental biological research and then also looking for ways to apply this research in a clinical setting. And I wonder how far you thought this most recent project that you presented was from clinical adoption, perhaps, or yeah, I think that's probably my key question.
For sure, yeah. So I would say the techniques we're employing in general, CyTOF mass cytometry for doing large-scale profiling of immune composition, hematopoietic composition, there are certainly efforts ongoing. I can tell you firsthand at Stanford right now, where some of my old trainees and some faculty at Stanford that are on the diagnostic side are actually working to leverage these multiplex technologies to like do a better job of existing clinical diagnostic workflows, right? Because there's a lot of things multiplexing brings to the table. The metabolism stuff itself, this is pure research right now. maybe there'll be something where someone will think of a cool way to use this to kind of go after metabolic disorders or disease. I don't know. I mean, maybe it could. But right now, this one's kind of research fun. We are certainly using it in a lot of kind of immunotherapy, immune modulatory biomarker discovery experiments where we're looking to see if there's weird dysfunction that's, dysfunction function that's associated with good or bad outcomes. So maybe that might lead to a diagnostic, but right now, like I said, it's just worth the fun stage. It's just like, what's a question we can ask about metabolism that's few enough cells or a weird enough thing to capture that hasn't been looked at before.
Gotcha. Thank you again for a wonderful talk. Really appreciated it.